Welcome. As I remove my mask this evening, I'll share that I'm joining you from my home in Blacksburg as we take part in this virtual event. I'm Jenny Rittenauer, Director of Alumni Chapter Programs at Virginia Tech, and it's my pleasure to welcome you to our Spring 2021 Hokies for Good event, Conquering Cancer Through Collaboration. We're excited to introduce this program as our second event under the Hokies for Good theme. For many Hokies, Prosim is more than a mindset, it's a lifelong calling. You'll find these experts and advocates tackling some of our world's greatest challenges with passion and perseverance. They're a positive force for change, and that's why we're celebrating them. Hokies for Good is a series of conversations and events with those who have dedicated their careers to humanity's most pressing issues. Tonight's program is no exception and the perfect fit for our theme. We jumped at this opportunity to get a front row seat to learn more about the partnership between Virginia Tech's Fralin Biomedical Research Institute at Virginia Tech Carilion, Children's National Hospital, and the Virginia Maryland College of Veterinary Medicine. I'd like to introduce Mike Friedlander, Executive Director of the Fralin Biomedical Research Institute at VTC and Virginia Tech's Vice President for Health Sciences and Technology. Dr. Friedlander is also the Senior Dean for Research and a professor at the Virginia Tech Carilion School of Medicine. Among his honors and recognitions, Dr. Friedlander is an elected fellow of the world's largest scientific society, the American Association for the Advancement of Science, a recipient of the William Menninger Award for Distinguished Contributions to Mental Health Research, and a Distinguished Scientist of the Society for Experimental Biology and Medicine. Dr. Friedlander has recruited some of the world's most creative biomedical scientists to Roanoke, leading the city's storied transformation from train town to brain town. He's also helped lead the efforts to develop the university's COVID-19 testing capabilities. With that, I will turn things over to you, Dr. Friedlander, to get the conversation started.
Great, thank you very much, Jenny. I'm in my office alone, so I'll take my mask off and get a little easier to hear me, I hope. It's a great pleasure to welcome everybody here this evening. It's always a matter of pride and excitement to be able to talk about Hokies doing good. There are lots of them doing a lot of good. Tonight's program will center on advances in cancer research and particularly related to collaborative efforts. I think, as everybody probably knows, cancer is indeed one of the leading causes of disease and mortality in the United States and the world, and numbers are increasing in many cases. There's a pressing need for new diagnostics, new therapeutics to treat cancer in children and adults throughout the lifespan. We're extremely fortunate here at Virginia Tech in that we have a number of really outstanding cancer researchers from across the university, not only here on the Health Sciences campus in Roanoke, but of course on the main campus in Blacksburg. And we're also extremely fortunate that we have this wonderful new partnership with our colleagues at the Children's National Hospital in Washington, D.C., many of whom are involved indeed with cancer research, in many cases focusing on pediatric cancer. So to really highlight the kind of collaborations that are going on between the different groups tonight, we've asked three people to join us and talk about their work. The first is Dr. Kath Bullard from Children's National Hospital, who I'll introduce in a moment. And Kath has an MD, a medical degree. The next is Dr. John Rossmeisel from the Blacksburg campus of Virginia Tech. And Dr. Rossmeisel is a veterinarian, has a DVM degree. And then the third is Dr. Ji Sheng from here on the Health Sciences campus in Roanoke, and Dr. Sheng has a PhD degree. So as I've emphasized, we have people that come from different aspects of training. And one of the things we want to emphasize tonight is how this all comes together to position us at Virginia Tech with the College of Veterinary Medicine, with the Fralin Biomedical Research Institute, and with our wonderful partners up at the Children's National Hospital to converge on some things, bringing to bear leading technologies and expertise across the spectrum in a way that really differentiates us from many other academic health centers. So the three speakers tonight, as I said, will be started by Dr. Kath Bullard. Dr. Bullard got her medical degree at the University of Otago in Dunedin, New Zealand. She also has studied immunology. She has gone on to have a number of positions. Currently, she is the director of the Center for Cancer and Immunology Research at the Children's National Research Institute, and she directs their program for cell enhancement and technologies for immunotherapy. She also serves as a member of the Division of the Blood and Marrow Transplantation Unit, and she's a professor of pediatrics and microbiology, immunology, and tropical medicine at the George Washington University, where she also serves as director for their translational research and innovation within the GW Cancer Center. Before coming to Children's National, Dr. Bullard was at Baylor College of Medicine in Houston, Texas, a place I know very well. The next speaker will be Dr. John Rossmeisel, a veterinarian from Virginia Tech's College of Veterinary Medicine. Dr. Rossmeisel is professor of neurology and neurosurgery in the College of Veterinary Medicine. He also serves as the associate head of the Department of Small Animal Clinical Sciences in the vet school. And he is the Dr. and Mrs. Dorsey Taylor Mahan Professor of the Virginia Tech Board of Visitors. Dr. Rossmeisel, prior to this, was associate professor in the department. He did his residency there at the veterinary school at Virginia Tech. And then prior to that, he was a research fellow at Auburn University in the vet school. In addition to that, we have Dr. Z Sheng. Dr. Sheng is here at the Fralin Biomedical Research Institute, where I'm located in Roanoke. Dr. Sheng is an assistant professor here and one of our very active cancer researchers. He did his postdoctoral training at the University of Massachusetts Medical School in molecular cancer biology. And prior to that, he did his PhD in molecular and cell biology at State University of New York Downstate Medical Center. And prior to that, he did his undergraduate work, master's degree at Shanghai Medical College, Fudan University. And Dr. Sheng is also an entrepreneur who's spun off a company in cancer research as well. So each of these three individuals is really renowned in their own respect for their cancer research. As I said, they all come at this a little bit differently, but I think you'll be able to see where some of these convergence points happen and why this is such an exciting time for these collaborations. So we're going to have each speaker tell you a little bit about their work, and then we're going to have time for a panel discussion and questions. If you have questions you'd like to have answered, please enter them in the Q&A or the chat. So without any more ado, Dr. Bullard, I will turn it over to you to give us the first presentation.
So thanks very much, Mike. And yeah, you stole my thunder that I was from New Zealand, because often I try and make the audience guess where the accent is from. So, but I will put you out of your misery right at the start, so you're not guessing the entire talk. So Mike, who I've known, as he said, since my Baylor days, asked me to talk a bit about our center, and then especially the work we're doing in cell therapy, which is was considered breakthrough of the year in science a few years ago, and has really been on a real upward trajectory for cancer treatment. I do have some disclosures. I have set up 2 companies in the cell therapy space, and I will be talking about 2 of those products today. This is our structure at Children's National in the research arm. And so as you can see, we are broken down into 5 different research centers, but all cancer research falls under my bucket, which, as you heard, is the Center for Cancer and Immunology Research. And I report up to Dr. Batshaw, who's the director of CNRI, who reports directly to our CEO. Currently, Children's National is top 6 on US News and World Report for pediatric cancer. We currently have 50 faculty in our center, with about just under half being lab or research focused, with the rest being clinician researchers. Of those who are funded, 74% have federal funding, and currently we bring in just under $9 million a year in funds, most of which are federal funding for the Cancer Center. It's a very active faculty with a lot of publications, especially over the last 3 years. We also have a robust intellectual property profile, second only to our Sheikh Zayed Institute, which is very device-driven. In oncology, we have 3 main areas: leukemia, lymphoma, solid tumors, and neuro-oncology. And it is this neuro-oncology group where we have set up very strong collaborations between Virginia Tech and Children's National. We have on the clinical research side a lot of research consortium studies, as well as multi-site non-consortium studies, and the so-called jewel in the crown investigator-initiated studies, which is where these are studies that have come directly out of researchers at Children's National and are translated to the clinic. We currently have more than 100 clinical trials open. As you can see here, in the last 3 years, we have had very robust activity in our clinical trial enrollment, and we're in the top 98th percentile for the children's oncology group nationwide. Only 4 centers, and they're all much bigger centers, have more enrollments than us. We also are very active in education and training and have 2 NIH training grants, these so-called T32 grants. We also have very robust mentorship committees, grant writer and editor support, and we provide pilot funding for our junior and senior faculty to foster collaborations. And one of the highlights I want to show you of our pilot funding program is the one between Children's National and Virginia Tech. This was a collaborative research initiative that began through an agreement through the Fralin Biomedical Research Institute and Children's National's Office of Academic Affairs and Research. Mike and I co-hosted a retreat last year to kickstart this collaboration between the researchers at both institutions. We then subsequently put out an RFA for 2 pilot awards, and as you can see here, 2 pilot awards went to investigators where 1 PI was from Virginia Tech and 1 PI was from Children's National. So in this case, Dr. Lee and Dr. Campbell were from Virginia Tech, and Dr. Pei and Zhu were from Children's National. So then moving to our cell therapy program, which we've called SETI, which is not, if you Google SETI, you might find that it's for extraterrestrials, but it isn't. It's called Cell Enhancement and Technologies for Immunotherapy. I was recruited from Baylor in 2013 to build a cell therapy program. And effectively, when I came, there really was nothing there. And by the beginning of 2014, we had recruited a critical mass of faculty and staff. But it's important to know there really was nothing when I arrived. And by May 2014, we did have 7 faculty. We did have what we call a GMP facility, which is where we manufacture these cells to give to the patients. And we had initiated some clinical trials. Now we are very active. We not only have a cell therapy program at Children's National, but also at GW. We now have 20 faculty with over 70 faculty and staff, bringing in over $6 million a year in NIH funding. We now have gone from 0 manufacturing suites to 4, and we're working on extending that further. We have 16 active clinical trials where we have developed novel cell therapies in our program to treat patients with cancer and devastating viral infections. And with that, we've treated over 170 patients since we started the program. This is our new GMP facility where you can see very clean facilities for manufacturing these cell therapies. So overall, as I said, we've treated over 170 patients with over 16 protocols. So I just wanted to give you some highlights about what is the power of cell therapies. And I first want to start about what we call NK cell therapies, or natural killer cells, and T cells. So T cells and NK cells are your immune cells that are in your peripheral blood and in your lymph nodes, and they are the cells that will protect you from getting cancer and protect you from viral infection. And what happens is, as soon as a T cell has been trained and ready to kill a cancer cell or a virus-infected cell, it will live lifelong. So this is a video. Hopefully it works. So here you have a big brain tumor cell. These are all the little T cells. As you can see, they're recognizing the brain tumor cell. They are puncturing holes in it, releasing chemicals that are killing the brain tumor cell. And unlike antibodies, these cells will, as I said, live long term. And once they've killed that cell, they are now armed and ready to go on and kill another cancer cell and another cancer cell until they have no further targets to kill. So natural killer cells and T cells can do similar things at killing cancer cells. And we looked firstly in a preclinical model. This is a murine model of a devastating pediatric cancer called neuroblastoma. And we showed that our novel NK cell therapy allowed these mice to survive long-term much better than our previous generation NK cell therapy or the old school types, or no treatment at all. So we went from the bench to the mouse to the clinic, and we started with T cell therapies for very difficult to treat pediatric solid tumors. We started without brain cancers because we were concerned that these T cells may elicit side effects that would not be tolerable in those patients. However, we did treat it was about 15 pediatric patients, many of whom were on palliative care, had exhausted all other therapeutic options for the treatment of their solid tumors. And these were the press releases that were associated with this study that was published in the Journal of Clinical Oncology a couple of years ago. But as you can see here, there are some children living for very long periods of time in patients who really were not expected to have a life expectancy beyond about 3 to 6 months. Furthermore, we showed that the patients, after getting the T cell therapy, were living much longer than after, or did not have disease progression for a lot longer compared to their previous standard chemotherapy, where they were relapsing pretty early. And I urge you to go to Matthew's story. He is a little boy with Wilms tumor who was told he had no other therapeutic options. He received these T cells that we made for him. We basically took his blood in the laboratory, retrained his T cells to kill his cancer, and we gave them back to him. He had no side effects whatsoever. And this is a really remarkable video where he talks about how wonderful it was getting T cell therapy versus chemotherapy, which made him really sick. And he still relapsed with chemotherapy, but he is still going strong with the power of the T cell therapy. So we have, obviously a very robust T cell therapy and cell therapy program. We've extended that study I just talked about to pediatric brain tumors, but we also are very active at using T cells to target viruses. T cells are our best defense against viral infection if you already have the virus. So with this current pandemic, we hear a lot about antibodies. Antibodies will stop a virus in its tracks when it starts to try and enter the host. But once the virus has already infected the host, your best chance of clearing the virus is your T cell response. So in patients after bone marrow transplant who have very weakened immune system, they have no T cell immunity and have devastating effects from viral infection. And this just is showing some of our data to show very high efficacy when we give our T cells targeting these very common viruses in our patients, CMV, EBV, and adenovirus, to prevent and treat these devastating viral diseases after bone marrow transplant. And I urge you, this is another story of a girl who was received these virus-killing T cells after bone marrow transplant. She was transplanted for leukemia. She was very high risk for getting devastating viral infection. She was an avid dancer prior to transplant, and after her transplant and T cell therapy, she has gone on to do extremely well. She's back to dancing and has graduated and really living a very active and full life. So we have been looking at targeting multiple viruses, including influenza, RSV, chickenpox virus, and many viruses that really have devastating effects on our patients. We also have clinical trials using our T-cell therapies for HIV, and more recently for norovirus, which is a really devastating gastrointestinal virus that affects our immunosuppressed population. So I gave this talk just in early 2020, and we said, oh, well, if we're treating norovirus, we can use these T cells on cruise ships. But of course, that was at the same time when the first outbreak of coronavirus occurred on a cruise ship. And so it wasn't such a funny joke by then. And we pivoted very quickly to develop a T-cell therapy for SARS-CoV-2. And we published it at the end of last year. Again, there are several press releases regarding this work. But the bottom line is, this is the team who in March 2020, we were starting this process of generating these SARS-CoV-2 therapies. And we collected 46 donors who had recovered from COVID. You can see most of them had very mild disease. Most of them had antibody responses to the virus. But interestingly, not all had antibody responses. And it was only in those with very robust antibody responses did you see the best T-cell immunity. So these are really great donors that we can use to collect their SARS-CoV-2 T cells from and use them to give to our very vulnerable patients. I think it's really important for the community to know that while these T cells that we were generating from the patients who'd recovered from COVID, and more recently from patients or donors who have received the Pfizer vaccine, we do see good cross-reactivity with the SARS-CoV-2 variants, but we see terrible cross-reactivity with the common cold virus. So this really illustrates why this virus took hold, because most of us had really little to no immunity against this virus. So as I said, our plan is now to use this novel T-cell therapy, in particular to treat our immune suppressed patients who we have seen are real prolonged shedders of the virus. We know that, for example, the UK strain, which is currently prevalent in the US and is more virulent or more infectious than the Wuhan strain, arose from a patient with lymphoma who was highly immune suppressed, and he had harbored the virus for months and months. Over that time, the virus had mutated to this more virulent strain, and that's what we're seeing repeatedly in our highly immune suppressed patients. So by giving these T cells to these patients, we hope to stop this viral shedding and hopefully will stop the propagation of these mutant strains of the virus that are plaguing us worldwide at the moment. So this is my last slide, just the take-home message with the power of the T cells. We take the blood from the patient, collect the T cells, train them in the laboratory to kill cancer and or viruses, and give them back to the patient to cure them of their disease. It definitely takes a village to do this sort of work. I won't belabor everybody on this slide, but I do want to give a big shout out to Mike, because it's through collaborations like this with Virginia Tech that we will be generating and developing the next phase of novel cell therapies. So thank you very much.
Thank you very much, Kath. Fantastic. Well, a lot of exciting information there about therapy that's being done, as you saw, in human patients and kids. Talk about T-cell therapy. We'll come back to that in the discussion. So let's go on to the next talk now. Dr. Rossmeisel, you're up.
Thanks, Mike. So as Mike said, I'm a veterinarian that's trained as a neurologist and a neurosurgeon, and my clinical practice and research programs are focused on the treatment of animals with brain cancer. So I'm showing you this slide because neurosurgery is inherently a specialty that represents a marriage of life science, engineering, and computer and data science. And it was these institutional strengths that actually drew me to a career at Virginia Tech in the first place. So as I take this opportunity to show you a snapshot of my research, I just want to highlight that these accomplishments are only possible through extensive collaborations with faculty leaders in engineering, biochemistry, cancer biology, and computational biology at Virginia Tech and our research partner institutions. You'll hear a little bit later this evening from one of my collaborators, Dr. Sheng, who I work with to develop drugs designed to overcome chemotherapy resistance in brain cancer. So I consider myself a comparative neuro-oncologist. And neuro-oncology is the discipline that integrates the naturally occurring nervous system cancers that we see in animals into more general studies of cancer biology and therapy for the benefit of all species that are affected by brain cancer. The incidence of brain cancer is actually very similar in dogs to that it is in people. And the most common malignant brain tumor in both of these species is actually called the glioma. And unfortunately, this type of brain cancer is notoriously difficult to treat. So a major focus of my work over the past several decades has involved characterizing the molecular and genetic landscape of canine gliomas. And so by performing cross-species comparisons of the cancer biology between canine glioma to databases of human, adult, and pediatric gliomas, we've identified several key similarities in the mutational processes and tumor immune environment between these two species. And so, as Dr. Bullard had said, identifying commonalities or targets in the immune system can be really important to develop therapeutics. So collectively, these studies have shown us that canine gliomas resemble human gliomas at the genetic and epigenetic levels. And actually, at the most granular level, canine tumors are more closely resemble pediatric brain tumors rather than human adult brain tumors. These discoveries provided a foundation for us to actually discover treatments that can help dogs and people with brain cancer, and further justify the use of dogs with naturally occurring brain tumors as very faithful translational models to study and evaluate new brain tumor therapies. So we have evolved to develop multiple characteristics that protect our brain from damage. And the blood vessels in the central nervous system, as I'm showing you in the cartoon on the left-hand side, actually possess several unique properties that are collectively called the blood-brain barrier, which allow these vessels to tightly regulate the movement of cells and molecules between the blood and the brain. So the blood-brain barrier is key defense mechanism against disease-causing bacteria and viruses and toxins that may be present in our blood. So, however, the protective function of the blood-brain barrier also hinders the delivery of many potential helpful diagnostic and therapeutic drugs that will prevent them from getting into the brain in adequate amounts to be clinically effective. So a big aspect of my research is actually to develop techniques and technologies to overcome the challenges with drug delivery to the brain. And one of these methods is called convection enhanced delivery, which I'm illustrating in this video on the top right. So convection enhanced delivery bypasses the blood-brain barrier and involves insertion of catheters into the brain tissue and then directly infusing drugs into the brain. We also use ultrasound and electrical energy-based therapies to disrupt the blood-brain barrier. So by applying tunable electrical pulses called electroporation or sound energy called focused ultrasound, we can transiently make the blood-brain barrier leaky in areas of the brain that we expose to these energy-based fields. So these types of energies transiently cause that blood-brain barrier to become disrupted and thus allow drugs to pass into the brain at clinically relevant concentrations. These energy-based techniques are really particularly attractive for clinical use because they can be applied to the patient without a need for a surgical procedure. So you can apply these energy therapies through the intact skull and scalp without a need to open the patient's head surgically. So in this slide, I'm showing an application of how you can use precision magnetic resonance imaging guided focused ultrasound technology to translate open the blood-brain barrier in a dog. And so the top panels are showing you the brain before we apply the focused ultrasound, and the bottom panels, as indicated by the red arrow here, you can actually see this focal circular region of area where we've disrupted the blood-brain barrier and allowed a dye that would not normally permeate the brain to actually be visualized here. And so the premise of this is now we can deliver drugs that wouldn't normally cross the blood-brain barrier into the region of brain tissue that we've actually exposed to that focused ultrasound energy. So Virginia Tech's Fralin Biomedical Research Institute and the Animal Care and Research Center facility in Roanoke and Children's National are one of only a handful of institutions in the actual whole country that have access to this cutting-edge MRI-guided ultrasound equipment. And this equipment is now being investigated for use to treat multiple types of brain disorders, including cancer. And so Children's National is some of the clinical trials that Dr. Bullard talked about. Children's National is poised to start trials involving focused ultrasound and pediatric brain cancers. So another technology that we've actually developed, it's been shown that the physical properties and the design of the catheters that you use for convection enhanced delivery, so that blood-brain barrier bypass technique that I talked to you about, are crucial to the efficiency and the successful delivery of drugs into the brain. It's not quite as simple as just sticking a catheter into the brain and then turning on a pump to get the drugs. The environment of the brain is very complex. It's heterogeneous, different tissue types, and so fluid flow is quite complex in the brain. So we've actually designed a catheter system called the Convection Enhanced Thermotherapy Catheter System to improve convection enhanced delivery treatments. So our catheter is controlled and deployed with a complete robotic interface that's coupled into the magnetic resonance imaging unit in a hospital. So we can actually watch these treatments with using MRI imaging, which is very commonly used clinical imaging modality for patients that have brain disease. And then we can actually adjust treatment as necessary based on real-time feedback from the MRI. And so our catheter involves 7 individually controllable microneedles that are made out of hollow optical fibers. And so these optical fibers can actually they can deliver light by a laser energy, and they can deliver drug through the lumen of the catheter. And so by having these individually deployable microneedles, we can actually tailor the treatment to various complex tumor geometries, and we can achieve a broad dispersal of the drug to effectively cover the tumor and the surrounding tumor margins. Other potential advantages of this system is that we can use the system to slightly heat the brain tissue, because heating the brain tissue will actually further disperse that drug within the brain. We can also use light energy to activate light-sensitive drugs in processes called optogenetic or photodynamic therapy, or we can actually use the laser to heat up the brain tissue to a point where it actually will thermally ablate or kill the brain tumor. So this catheter can perform multiple different types of therapeutic interventions. So the principal mechanism by which I actually assess our research discoveries is through the conduct of clinical trials in pet dogs with naturally occurring brain cancers. And these trials frequently involve combining multiple innovations from the collaborators that I work with. And so the example I'm going to show you represents one of those collaborations. So we've identified 2 receptors that are present in both canine malignant brain tumors and human malignant brain tumors, called the IL-13RA2 receptor and the FA2 receptor. And these receptors are only present in cancerous cells, and so they're not present in differentiated or normal brain cells. So we've developed an engineered bacterial cytotoxin, so a bacteria that we've engineered and then we've actually tagged to a protein that will bind to these receptors and subsequently internalize that bacterial cytotoxin and kill the cancer cell that contains these receptors. So this clinical approach has been used before in people, but one of the major obstacles to the success of the trial that's been and was previously performed in people is that they had a lot of technical difficulties with the convection enhanced delivery method. So these are very large drugs that will not penetrate the blood-brain barrier, so you actually have to give it through a delivery mechanism, either through blood-brain barrier bypass, like convection enhanced delivery, or potentially blood-brain barrier disruption. So another part of this trial actually involved refining and improving the convection enhanced delivery method. And so we do that by performing individually patient-specific therapeutic plans, some using computational software that will help the neurosurgeon decide where they should actually place the catheters into the tumor to get tumor coverage. And then we actually use the convection enhanced thermotherapy catheter system that I talked to you about. And so on the top right-hand panel here, you can actually, and then we monitor the infusion in real time. So this whole entire surgical procedure is done with the canine patient in the MRI unit. And so you can see we slowly pump in the drugs, and the drugs are actually conjugated to an MRI imaging contrast agent. So we can actually visualize the drug distribution within the tumor. And so these static images show you in 3 planes over the time course of the infusion. You can see that white plume gradually enlarging. Sorry about that. And that represents the drug distribution within that tumor. And then we can actually do 3D reconstructions and evaluate the efficiency of the procedure. We can also watch these movies in real time. And so we can do 2D, so we can actually look at the axial plane of the image. You can see that white plume filling up the tumor here, or we can actually watch it longitudinally to make sure we cover the entire tumor. And so this is really important because it allows us to actually make determinations on the efficacy and the toxicity of the treatment, because we know exactly where the drug was distributed, both in if it's the normal brain is exposed or if the tumor was exposed. And so in this study, we actually successfully treated 17 dogs with no toxicities after escalating that dose of cytotoxin therapy 6 different times. Although this particular trial was not designed to evaluate the efficacy of the drug, we actually saw a remarkable and promising preliminary efficacy signal, which I'm summarizing in this slide for you here. So following a single treatment, we actually observed an objective reduction in tumor size in 94% of the dogs that we treated at the first post-treatment recheck. And then 50% of these dogs had a more than 65% reduction in their tumor volume that lasted for greater than 6 months. So to put that in perspective for you, a dog that's diagnosed with this type of malignant glioma without any therapy is typically only going to live for about 2 months. And so these dogs actually had profound reductions in their tumor volume, as you can see here, that lasted for a long period of time with no other treatment and after a single infusion of the cytotoxins. So this study demonstrated that these targeted cytotoxins can be safely and effectively delivered to dogs with gliomas at doses 6 times higher than were previously given to human patients. And these cytotoxins have objective and clinically relevant anti-tumor effects over a wide range of doses. So this kind of study provides crucial preclinical data to move and translate this therapy to the human clinic. And then finally, when comparative oncology really works, this is kind of what you'd like to see. So these are press releases that are simultaneously showing you that this cytotoxin approach has been licensed simultaneously for development and use for dogs with brain cancer, and also to develop clinical trials in humans with brain cancer. And that's parallel and simultaneous development pathways as a comparative oncologist is kind of really the holy grail that I'd like to see from all my research. And as Dr. Bullard said, this is actually, I'm heavily reliant on multiple collaborators, which are represented in this slide. And I thank you for your time and participation.
Thank you very much, John. That was outstanding to see how this important work is helping patients or animal companion patients as well as humans, and how those two tie together. We'll talk more about that coming up. So next, finally, on to Dr. Zisheng. Xi, you're up.
Thank you, Mike. It's my great pleasure to having this chance to join with Dr. Rossmeisel, Dr. Bullard for this panel discussion.
Hey, Z, you might want to go into presenter mode. We see in all your slides on the side there,
Sorry.
There you go. That's great.
Okay,
thanks.
Good. So I'm Jay Shen. I'm an assistant professor at the Friedlander Biomedical Research Institute. I'm also have a startup company called Selectite Therapeutics, which I'm going to talk about a little bit about today. This is my disclosure. I do have financial interest in Selectite Therapeutics Incorporated. And also I will talk about a little bit with the collaboration with Dr. Rossmeisel that involved a drug that developed by First Strain. So you have to listen to a fantastic talk from clinical doctors. So I'm give you a little bit introduction of my career path, because I'm going to switch the topic to basic science. So I came from China. I'm a pathologist in China, but I found that my major interest is still research. So I went to USA. I became a molecular cell biologist. And then I did my postdoc training as a molecular cancer biologist. After Dr. Friedlander recruited me here to Roanoke, I had my own laboratory with a focus on translational cancer biologists. So most of the work I presented to you today is still bench work, but we hope that in the future we could moving toward bedside. So my lab primarily focuses on this we call precision medicine or targeted therapy for cancer. So I will give you a brief introduction what we are doing, and then I will focus on two specific projects that we have working on. One of which is a collaboration with Dr. John Rossmeisel. So my lab are interested in identify new and effective drugs that can be used to treat glioblastoma, the most lethal of brain cancer. You have already heard from Dr. Rossmeisel about this disease. So we are using different genetic tools to screen the genome, like 20,000 genes, to identify the genes that are suitable for the drug target for glioblastoma, with a major focus on the genes that could be very important for the unlimited growth of cancer cells. And then once we have those candidate genes, we are going to use biochemical and structural analysis to understand how this gene works in cancer cells. And further, we're going to study how these genes working on specific cells called cancer stem cells. The reason why I bring this up, which I'm not going to talk about today, but I want to bring this up, is because this is the basis of targeted therapy, or we call precision medicine for glioblastoma, because the existence of those cancer stem cells in brain cancer. And we want to eliminate all those cancer cells so that we will be able to effectively treat glioblastoma. And lastly is the, based on all these findings, we are going to develop cancer drugs to treat glioblastoma. So this is the story that I'm going to talk about later in a few slides. We are going to try to develop a new peptide medicine for glioblastoma. So we have worked on glioblastoma for many years already. I will make the long story short. So we identified one target called P110 beta, and this P110 beta, we found that this P110 beta is highly expressed in glioblastoma patients. And if a patient has high level of this protein, it will live much shorter than the patient with low level of this protein. That means this protein is very important for the tumor to grow and to develop. And we further, we use commercially available inhibitor of this specific protein. We found that it stopped growth of the tumor in animals. Even though this is not very, very promising, but it suggests that this is a cancer, brain cancer target. However, we did have a lot of challenges when we make a decision to working on this cancer drug target. So what is P110 beta? P110 beta is a signaling molecule. It has three other very similar siblings. And what they do is they convert a lipid called PIP2 into PIP3. And that PIP3 will further activate all these proteins to promote cell growth and promote cell dividing in cancers. So it's a very important signaling pathway in cancer. So one of the challenges that we are going to facing when we decide to work on this protein is the big pharmaceutical companies have already working on this pathway for many years, and they have developed a lot of inhibitors. Some of them have already in clinical trials. So we're kind of competing with the big pharmas to working on this candidate drug target. And the second challenge is, as I told you earlier, that they have four very similar proteins to each other. So when you look at all their 3D structures, they look very, very similar. And I will come back to this point later. The third challenge is if we try to use it as a drug target, we may need to figure out whether it has a pharmaceutical market for the future medicine that we're going to develop. Indeed, there is a market for the peptides, which I'm going to talk about, the peptide inhibitors. Even though compared to the small molecule drugs, it is a very small market. However, it increased. We know that this is challenging, but still encouraging us to continue to work on this drug target. Now, this is the most challenging part of this study. So, as I told you earlier, those four proteins, they are very, very similar to each other. So those proteins are composed of what we call amino acid. And the amino acids in this four protein, they are almost 98%, more than 98%, they're identical. So it's represented here by red bars. They are almost identical. And the gray bars means they are different, and less than 2% of these amino acid sequences are different. So it's like we are trying to find the difference between those four proteins within the 2% of the amino acid sequences. And we're lucky. We found a motif we call beta 18 motif, which is 18 amino acids only found in this P110 beta, but not other 3 siblings. So based on this finding, we developed a new peptide drug we called Selectide 18. So Selectide means selective inhibitory peptide for P110 beta. 18 means they have 18 amino acid. So what does this peptide inhibitor do? So we predict that this peptide binds to the protein called P85 alpha. So P85 alpha forms a complex with P110 beta. They form a signaling complex so that they are functional. What we found is that the Selectide 18, which binds to P85 alpha, will prevent the formation of signaling complex of P110 beta and P85 alpha. So this is a specific and selective inhibitor of P110 beta, but not alpha, delta, and gamma. We have the experiment showing that, which I'm not going to show you here, that this Selectide 18 only block P110 beta without affecting alpha, delta, and gamma. So why this is important? So basically, what we found is that glioblastoma relies on beta to grow, to survive, to develop, and they don't need alpha, delta, and gamma. So this provides some therapeutic advantage that we can only block beta so that we could selectively target it to glioblastoma cells without affecting normal cells. So we did an animal experiment to prove that whether this is a drug or not. So our Selectide 18 could reduce the tumor volume over time, and the control tumor still continue to grow. So we do have the strong evidence showing that we have a new peptide inhibitor for glioblastoma. Now, my lab really just focus on the cancer, particularly brain cancer. So our peptide drug can be absolutely used for as a new treatment for glioblastoma. However, the P110 beta not only important for glioblastoma, it is also important for a bunch of different cancers, such as breast cancer, lung cancer, ovarian cancer, and leukemia or lymphoma. So this drug not only can be used for glioblastoma, but also can be used for other cancers as well. Now, PI3K signaling, which is involved these 4 proteins, they are not important for important for malignant disease such as cancer, but also for those non-malignant diseases. So our drug also perhaps have can be used for this disease as well. So because of this, we have started a new biotech company we call Selecta 18 Therapeutics. Our slogan is we try to develop next generation peptidic medicine to treat human disease. So that's the one of the story that we're going to talk about. This is the second one. I want to focus on the collaborations, which many focus on the collaborative research with Dr. Rossmeisel and other faculties at FBRI. So this research started with the collaboration with Robert Gordy and also Gary Siemens at the Canadian Clinic. Robert Gordy is a director of our Center for Heart and Reparative Disease. He is expert of cardiovascular disease, and he really focused on a protein called connexin 43. So we worked together, and we found that connexin 43 is important for glioblastoma cells to become resistant to chemotherapy such as temozolomide. And Rob has developed a new peptide inhibitor for connexin 43, which is already FDA approved. And we use his FDA approved drug. And we found that if we combine this drug, ACT1, which is a connexin 43 inhibitor, and temozolomide, we could substantially reduce the tumor volume of brain cancers. So based on this research, we also collaborated with Sammy at FBRI, a cancer biologist at our institute, and Johan Foster at Virginia Tech. He's a chemist. And we generated a nanoparticle conjugated ACT1 peptide. And this purpose is for slow release so that we can apply to patients. So then we collaborated with Dr. Rossmeisel, and we studied a phase 1 clinical trial. And what we want to test first is that whether we can deliver this nanoparticle conjugated ACT1 peptide safely into the brain of canine patients using convection enhanced delivery, the method that Dr. Rossmeisel just described in detail. So this is all the research that I want to talk about today. And however, I want to thank all the lab members, past lab members, and our current lab members. Without their help, we will not be able to make all these breakthroughs. That's it. Thank you for your attention.
Thank you. Thank you, Z, very much. And thanks to all 3 of our speakers. So we have a little time for questions. I'd like to throw out a question to all 3 of you. Every one of you described what I would call interdisciplinary team science research. How important is it, and briefly from each one of you, how important is this process that you have access to other researchers that have other skill sets and different technologies, et cetera? And why is it so important to the work you're doing on cancer? Why don't I just go to each of you? Kath, what's your feeling about that?
So, for the sort of bench to bedside work that we do, and I noticed one of the questions in the chat is how were you able to translate novel therapies so fast from the bench to the bedside? And that really is purely and simply because of team science, partnering with PhDs, MD PhDs, and clinicians to really make that transition happen from mouse to man.
Yeah, good. Thank you. John?
Yeah, I wholeheartedly agree with what Kath said. It's fundamental not just to get those therapies to testing in patients, but also to refine those therapies as you go through that development process and go from I'll say the animal clinic to the human clinic. So it's an iterative and evolutionary process that requires a lot of teamwork.
Right, G.
Yeah, I want to echo what Dr. Bullard and Dr. Rossmeisel has just emphasized. So without all those different expertise, we will not be able to do that. So even though my lab focuses on precision medicine, we want to develop targeted therapies. We have to collaborate with, for example, expert with bioinformatics. So because we want to analyze the patient data. And also the clinical doctors, we want to obtain all those tumor samples so we can analyze. And also in the future, if we have new therapies, we need to collaborate with clinical doctors to convert this into the bedside treatment.
Okay, thank you. Another common thread I heard from all of your talks I'd like you to comment on is each of you has either a partnership with private companies, industry, pharmaceutical companies, or have spun out companies, for example, and you all work really in either academic institution or academic hospital environment. Why is that an important part of the partnership to actually get things out there and help patients, whether they're humans, children, adults, dogs, whatever it might be? John, why don't I start with you this time?
Yeah, it's important to have industrial and governmental partners because to actually scale up these things from clinical trials to actually treating the patient population that's in need, we need infrastructure oftentimes that can only be provided by some of these larger companies and experience. So it's really important to have those. Again, just that to me is just another representative level of collaborative science. So
fair enough. Kath?
Yeah, so, for me, because we're developing living drugs where you're training a patient's own immune system to kill cancer, it really didn't fit the pharma model. And I was, for 20 years, developing these new living biologics, and they'd go to phase 1 and then not go anywhere. And now the field really has got traction in the pharma space. And I realized, we just shouldn't be developing therapies for them to die in phase 1, even when you're seeing efficacy as well as safety. And so without starting your own companies and sort of partnering with biotech, you will not get beyond just boutique centers. And to make the greatest impact, you have to partner with industry.
Yeah, and maybe before I go to Z on that, Kath, you could comment. I know Children's National, of course, has this wonderful new research innovation campus in Washington, D.C. And as part of that, you have a partnership with Johnson & Johnson, their J Labs up there. How does that opportunity, create these sorts of connections you were just talking about for you and the other researchers there and the Virginia Tech researchers who will be up there?
No, I think that's right. For the Virginia Tech researchers and for the Children's National researchers, having that sort of incubator, if you like, with these sorts of pharma companies, J&J being the biggest, this will be really a great opportunity for many of us to see our new developments at the bench, realize in the clinic, and not just in a small phase study, but really going beyond that. And so it is a real opportunity, and so we are excited for you guys to join us as a result. Yeah, for sure.
Yeah, likewise here too. And G, you want to comment on the commercialization, company partnership, spin-off aspect of its role and the importance of this research?
Yeah, as I told you earlier, we started a company, and also I showed you that the pharmaceutical market for the peptide drugs compared to the small molecule drug. So the big pharma, they always favor those small molecule drug, but not the peptide drugs. So that's the reason why we started our own spin-off company to try to translate our research into the clinic. In the future, we do need to partnership with big pharma so that we can move this forward.
Okay. John, there's a specific question for you in the chat. They wanted to know about the size of your catheters that you're using.
Oh, so yeah, those catheters are really small. So the internal diameter of that catheter is about 250 microns of the lumen. So it's pretty small stuff. And so the outer diameter is about a 0.5 millimeter. So these are pretty small catheters that we're putting in. Yep.
And I'd like to ask any of you or all of you to comment on this. You gave great examples in all three talks about this particular relationship and partnership that is within Virginia Tech from the College of Veterinary Medicine and here at the Health Science Center, for example, and at Children's National, the work going on there. And so this whole kind of nexus of research that's targeted primarily, but not exclusively, at pediatric populations for Kath and her team, but then also here at canine patients and other companion animal patients. How unique is this relationship with specific, focus areas, the similarities in some of the tumors that you described? Are there a lot of places that have all this together and coming together? Is this unusual or unique? And what are the advantages? How do any of you see that? Anybody can take that.
I mean, from my point of view, it's extremely unique. I think the partnership, especially with the veterinary component, is really very, impactful. Then plus with the imaging that Dr. Sheng talked about, and the pediatric population that we serve, which is not only obviously pretty much all pediatric cancer, but a very diverse patient population and very unique. And so this is truly a very special partnership and one that's very dear to me as well.
Yeah, Mike, there's only a couple of dozen veterinary schools in the whole nation. And, some other ones definitely have comparative oncology programs, but, the focus area we have here at Virginia Tech is quite unique for a number of different reasons. I think Virginia Tech is probably, not only on the pharmaceutical or drug side of things, but Virginia Tech is in comparative oncology is definitely a very unique an innovative center for cancer devices, so biomedical devices for cancer treatment, for sure.
All right, thank you. All right, well, go ahead, G. Go ahead. I'm sorry.
Yeah, I completely agree with you. So just for example, for my own research, and I want to do more cancer research, and also the same thing for other cancer researchers. We're not only just trying to develop the cancer treatment for human patients. It also can be used for other patients as well, particularly because my lab focuses on the adult brain cancer. But now with this partnership with Children's National, we are creating more projects that we're going to work on the pediatric brain cancer.
That's great. In so many cases, the pediatric population doesn't get as much attention as the adult population in research. So that's a wonderful aspect, I think, of this partnership. All right, we're just about out of time, but I'm not going to let you get away without giving you each a chance to tell us your idea of what's the next big thing in your own cancer research or cancer research in general. What should the audience and everybody else be looking forward to that's coming down the pike that you're particularly excited about? I'm gonna, Xi, I'll let you go first this time.
Sure, yeah. I think what's the most exciting thing about the future of cancer research is precision medicine. And the reason is we have to treat cancer patient differently, each individual patient differently, using completely different method. And now we are in the era of genomics, proteomics, and we can do a lot of big data analysis with all these tools together. In the future, I believe that we will be able to treat each individual patient differently, effectively, and using precision medicine.
All right, thank you. John, go to you for the next one.
Yeah, I think, building on what G just said, I think we're going to be combining those precision medicine approaches with really new and novel ways to get drugs into the brain that isn't going to require surgery in the traditional sense, like that focused ultrasound technology that I talked to you about. So non-invasive ways to actually get beneficial therapeutics into the brain without actually having to do a conventional surgery on a patient. I think that's going to be it. And then, for brain cancer, I really think it's going to be some combination of some immunotherapeutic approach, largely like the type that Dr. Bullard is working on. That's going to make a big difference. So,
okay. good setup for Dr. Bullard.
Yeah.
Kath, you want to take the last?
So you have to have some context. I got into cell therapy because my best friend in high school got Hodgkin lymphoma, and she was cured of her Hodgkin lymphoma, but died of leukemia. That was the direct result of all the chemotherapy and radiation she had received to cure her of her Hodgkin lymphoma. So my hope for the future, and I do think it will be realized, is that we will not need chemotherapy as we know it today, and we will not need radiation as we know it today. And so it will be targeted, it will be precision medicine, and it will be immunotherapy.
That's great. Well, those are all very exciting opportunities. Before I thank the speakers and the audience and close, I just want to say for those of you who haven't seen yet, of course, in the pandemic year, people haven't been traveling much, but we just have an enormous opportunity here. Virginia Tech's growth in the health sciences, including a big focus on cancer through the Cancer Research Alliance that spans the campuses, the new facilities here in Roanoke, what's going on in the College of Veterinary Medicine, their oncology center here, and our partners at Children's National Medical Center in Washington, D.C., that have this wonderful new facility that's at the former Walter Reed Army Medical Center campus, where we'll be able to share some space from them. If you get a chance, visit any of those places. It really represents a kind of a coming together in physicality to the nature of this collaboration. But more important than the physicality, of course, is the people and the ideas that are permeating and connecting us together. So I think you're going to hear a lot more exciting things coming out in cancer research from these groups. So to the audience, thank you all very much for joining us. To our speakers, Dr. Rossmeisel, Dr. Sheng, and Dr. Bullard, thank you very much. It was really a wonderful and exciting evening tonight. Thanks again.
Thank you.
Thank you so much for having us.
Thanks, Mike.
Bye.
Thanks, Joel. That was great.
Thanks.
night.
Bye.